So welcome everybody. I'd like to introduce Dr. Yang Su. He's a new technology manager with Microfluidics International Corporation. And he's going to talk about creating nanoparticles with Microfluidizer high shear fluid technology. And really he's going to tell you what to do if you don't like your particle size distribution. So go ahead and take it away, Yang.
So as Jeff already pointed out, I'm going to talk about how using Microfluidizer high shear technology can help you creating nano-sized particles. So for those of you who are not very familiar with the microfluidics and the Microfluidizer processors, I have a brief introduction to our company and technology at the beginning. Then I'm going to spend the majority of the time to talk about different applications that different type of nanoparticles you can create using our technology. So microfluidics has been around for over 30 years. We're located just outside of Boston. Microfluidics is the manufacturer of Microfluidizer high shear fluid processors. We have sold thousands of processors to our customers worldwide. The Microfluidizer high shear fluid processors really offer unique technological advantages that can help you produce nano-sized particles with a variety of multi-phase applications. We have a full line of instruments that runs from small lab scales all the way to the large production scales. And our customers are actually using microfluidizers for a variety of applications in different industries, such as biopharmaceutical industry, chemical industry, nutraceutical and food industries, as well as cosmetic industries. What microfluidics or microfluidizer does best is we can create a variety of nano-sized materials, such as nanoemulsions. We can do very high efficiency cell disruptions with very high protein recovery. We can reduce particle size efficiently. And create the particles that can encapsulate valuable materials inside, and we can efficiently agglomerate the dispersions. Today's talk, I'm going to primarily focus on the biopharma industry. So most of my applications will be within that category. So at the bottom, you can see a different type of microfluidizers. On the bottom left here is a lab scale benchtop machine, which is the M110P unit. The next one shown here is our pilot machine, and this one on the right is our M700 series, which is the production scale microfluidizer. All of these machines are using the same core technology, which is our fixed geometry interaction chambers. I'm going to explain that just in a minute. So first, I want to show schematic that how microfluidizer works. Generally, we have an inlet session where you can feed your materials into the microfluidizer. The microfluidizer is designed to be operated either in a batch mode or in a continuously fashion. The machine is robust enough to accommodate very high solid contents, and also it can process materials with high viscosities, as well as a wide range of temperatures. The materials will be put in by an intensifier pump. The intensifier pump is capable of delivering very high pressure, up to 40,000 psi. And also, it's delivering this pressure at a constant pressure rate. So the material will be pressurized by the intensifier pump and pushed through the fixed geometry interaction chamber where the pressure energy is changed to kinetic energy, resulting in very high velocity, which gives very high shear and impact forces. Also, there's a very high level of turbulent energy dissipation rate occurs, which all helps reduce the particle size. Following the interaction chamber, there can be the product temperature can be controlled by using a heat exchanger. Like I said, our core technology is the interaction chamber. We have two types of interaction chamber designs. The one showed on the lower left is the white type interaction chamber, where the inlet fluid is actually split into two streams and pass through microchannels and form an impingement flow in this impact zone, where there's a very high degree of energy dissipation will occur. So the white type interaction chamber ideal for liquid-liquid type of dispersion, such as nanoemulsions, liposomes, or polymer nanoparticles. The interaction chamber showed on the lower right is the Z-type interaction chamber, where there's only one microchannel, and there's also a post-channel liquid-to-chamber wall type of impact area. The Z-type interaction chamber is ideal for processing samples that contain solids, such as doing dispersions or particle size reductions, as well as cell disruptions. The unique design for the interaction chamber is there's no moving parts, so the geometry is fixed, which guarantees the scalability. Also, the interaction chambers are made with very hard materials such as diamond or ceramic, which offers excellent wear resistance and chamber life. We also have different types of shapes and size for the microchannels, which give you a variety selection of shear rate. Like I said, so how we scale up is we actually add more identical microchannels in a parallel fashion. So each microchannel will be the same as a single channel, as long as you keep your pressure, maintain your inlet pressure. All the velocities, shear rate, and energy dissipation occurred inside each microchannel will be identical, which means when you scale up, this is often we're calling a direct scale up. You can just dial in your process conditions you develop on the lab scale machine directly to the production machine, and you will get the same results. Here I want to show the difference will caused by pressure profile. In Microfluidizer, like I said before, we deliver a constant pressure, which is shown here with the green line. You can see during one cycle, the majority of the time, over 90% of the cycle time, the pressure is maintained at the target pressure, which means that majority or virtually all the product will be processed at this pressure. This ensures you get very tight particle size distribution. While some conventional technologies, such as high-pressure homogenizers, actually give you a variable pressure. You can see with the right line showing here, during one cycle, you actually only have very minimum time that pressure will be maintained at the target pressure. The pressure actually varies from this target pressure all the way down to the half of the set pressure. So you can imagine that the material will be processed at different pressure, which will result a bigger particle size, or in other words, a board size distribution. This just shows the shear rate we can achieve by a microfluidizer compared to some of the conventional technologies. On the left here, you can see some of the low energy devices, and here's a high energy devices. Microfluidizer can achieve shear rate orders of magnitude higher than other conventional devices. which gives you the best results. This is what I said before. By working with the different microchannel size and shape, so combining with the pressure, we can achieve a full range of different shear rates. So we'll be able to provide a solution for your unique applications and material requirement, because each application or material may require a different shear rate to achieve the best results, and we're able to deliver that shear rate for you. Just to summarize the unique benefits for microfluidizer processors, our technology is unique because we can deliver constant pressure. We can achieve very high pressure processing pressure. We use fixed geometry interaction chamber. When we scale up, we use multi-slotted interaction chambers. And these unique designs or features will benefit our customers with potentially very small particle size, with very narrow size distributions. And also we guarantee the scale-up results. A quick example shown here is this is the LV1 microfluidizer, which is our lab unit can handle the smallest amount of sample size, as small as 1 milliliter. You can develop all your formulation and process condition with this unit here, and when you scale up, you can scale up directly to a production scale microfluidizer. In this case, this is a M7250-20 CP machine. This machine is capable of delivering flow rate at up to 8 liters per minute and 1,300 bars. The last that I want to mention is all of our pilot and production machines are designed for compliance with cGMP and our CIP and SIP capable. Now, I want to move to my first application, which is nanoemulsion. Nanoemulsion, as many of you already know, are dispersion of 2 or more immiscible liquids with one phase finally dispersed in the others. Depends on the which phase is the continuous phase or which one is the dispersed phase. We can usually characterize the emulsion as oil in water emulsion, or water in oil emulsion, or multiple emulsions, or double emulsions, such as oil in water in oil, or water in oil in water emulsions. Surfactant is generally used when formulating the emulsions because the newly created nano-sized droplets need to be stabilized to prevent them from recoalescing to form back to the large particles. There's a variety of surfactants are available for selection. The examples I'm going to give today are mainly oil in water type of emulsions because these emulsions are very useful for acting as a drug delivery systems for delivering water-insoluble drugs because most of the newly developed drugs have poor water solubility, so they can be dissolved in the oil phase and encapsulated inside the oil phase and form a nanoemulsion for delivery. Nanoemulsion also has another very interesting characteristic, interesting property that some of the nanoemulsion formulations have shown to improve the immune response. So they can be used as vaccine adjuvants. I'm just going to give a very brief description here. Actually, if you're interested, there's a very good review articles you can find in this place here. So vaccines, as we know, are prepared to prevent a pandemic of a disease. The recent development of subunit vaccines are really improve the safety of the vaccines because they don't use the entire virus. But by doing that, the immunogenicity is actually greatly reduced. So this vaccine has to be paired with an adjuvant. The role of the adjuvant is to enhance the efficacy of the vaccines. For a very long time, alum is the most popular adjuvant that being used. The research have found that alum adjuvant primarily introduce Th2 type of immune response. However, some of the novel nanoemulsion or liposome vaccines adjuvants have shown that they have the capability to introduce not only Th2 but also Th1 type of response. This makes them very interesting adjuvants because they can be used for vaccines that are targeting disease which require Th1 type of response. Also, the formulation of nanoemulsions or liposomes can be adjusted so they're very well tolerable by human body, and biodegradable ingredients can be used. This slide tries to show why achieving a certain particle size and measuring the particle size is important for the production of nanoemulsions. Because the particle size will not only affect the stability of the nanoemulsions, they will also affect some of the post-processing processes, such as sterile filtration. In terms of stability, we know that presence of particles over 1 micron in the size may destabilize the emulsion through the so-called outward ripening phenomenon, where the large droplets will absorb the smaller droplets, and so they become larger, even larger droplets, and this process will go on and until the emulsion is unstable. So the process really need to be controlled that not produce many or any large particles, and also your particle size analysis method should be able to capture this. The other thing which is important, because a lot of the nanoemulsion formulation are actually administered through injection, so most often a final sterilization process is critical and required. Due to the limitation of formulation, sterilizing with heat is not always an option, so filter sterilization has to be employed. During the sterilizing filtration process, the filter membranes it generally has a process rated at 0.22 micron or 220 nanometer size. Here I have I'm showing two examples of different membranes. The cross-sectional view on the left is an asymmetric membrane where your process is actually varying from large to small. On the right-hand side, this is a symmetric membrane. Also is 0.22 micron rated, and you can see even it's set as symmetric membrane, which indicate has a more or less uniform process distribution. You can see the processes are actually not uniform. Some are big, some pores are small. So which means you have to control your particle size so the droplets, the majority of the droplets can be easily penetrated through different pore sizes. And you can imagine that because the membrane is rated as 0.22 micron, any particles larger than that can potentially clog the filter. Here comes the first example of an adjuvant nanoemulsion. This is the squalene-based nanoemulsion. Showing the results here, the black curve is the unprocessed, which shows large average particles. And after processing, the results actually shown here are processed by two different microfluidizers. The blue and purple curve are actually processed by a pilot scale M110EH microfluidizer. The green and red curves are actually processed by a larger scale production microfluidizer. You can see after one pass, the results are really close, and it's get even better after 3 passes. Both machines are actually giving identical results. This proves that our scalability from pilot scale to production scale. Because of the size are too small, usually around 100 to 200 nanometers, most people usually use dynamic light scattering method to measure the particle size. But we do encourage people to look at the laser diffraction measurements as well, because the volume-based measurement can sometimes give you information on the larger particle size. So if you look at here, you can see the small bump here. That means there may be a detectable particles or droplets very large. So if you want to, your emulsions can be passed through the sterilizing filters, you definitely want to get rid of those particles here. So we also recently did a study, it's a collaborative study with Paul Corporation. We look at the filtration results with very different size of nanoemulsions created by microfluidizers. By adjusting the process conditions, we're able to achieve nanoemulsions with different particle size. And here shows the mean particle size and from as small as 124 nanometers all the way to 170 nanometers. And you look at the filtration flux results, you can see the highest, of course, the smaller the particle, the higher the flux you can achieve. So the highest flux is achieved at the smallest mean particle size at 124 nanometer. You can see here, although your mean particle size are well below 200 nanometers, as small as 150, you're still not seeing a very high filtration flux, because you may have particles above 220 nanometers, which may slow down your filtration process. And also, when you look at the particle size before and after, this is for the case of close to 220 nanometers. When you look at the results before the filtration and after filtration at 2 different pressures, you can see virtually there's no changes in the particle size. This indicates that the filtration did not change the droplet size. The filter membrane did not change the droplet size. Also, there's no indication of losses of the oil phase during the filtration process. Next example for nanoemulsion is actually our castor oil nanoemulsion used for ocular delivery. Again, this showed results from 2 different microfluidizers, the pilot scale and production scale. All results are really close. You can see the production scale actually gives slightly better results than the pilot scale. The reason of that is this production unit here, 7250, it actually has 2 intensifier and can be worked as a way to deliver constant pressure, true constant pressure. So you won't have to see the dip of the pressure during processing. So this sometimes give you additional benefits than single intensifier pump. Also, we looked at different pre-mixing preparation method here. Some of the samples are prepared with the rotor-stator mixer, and some are just prepared mixed with the low-energy propellers. I do want to point out here that the pre-mixing step is a critical step that will often determine your final particle size and size distribution. However, in this case, we didn't really find much of they're slightly different, but differences are small. But in some applications and processes, this can be critical. The last example for nanoemulsions actually formulated with soybean oil. This is anesthesia nanoemulsions. Again, the optical microscope pictures show here. You can see the large droplets with the unprocessed samples, and the fast reduction after microfluidization just after 3 passes at 20,000 psi. The particle size are reduced greatly. The D90 is 240 nanometers, and this is the indication that this at this stage the emulsion may be able to pass through the sterilizing filter pretty easily. The next application is cell disruption. Cell disruption is actually a very important step for anyone using the cell-based expression systems. This has been the cell-based expression system has been used for a long time, and it's a mature technique for getting important proteins, enzymes, or any other products. And anytime you need to harvest the product from a cell, or inside the cell, you need to rupture the cell wall so the product inside the cell can be released. The rupture process is actually want to achieve recover the most of the product by number 1, rupture the highest percentage of the cells. Number 2, at the same time, to minimize the damage, such as the nature of the protein, damage to the product, either by shear or temperature or any other factors. Microfluidizer is a well-known technology within the biotech industry. I'm going to show you just in a minute. Remember, I said we are able to deliver a variety and a controllable shear rate. And this is very useful because if you look at the bottom here, that the shear rate required to rupture the cell walls for different types of cells, you can see the mammalian cell is actually pretty easy for to be broke. You only need low pressure and low shear to break those cells. Bacterial cell is actually a very commonly used cells, and they will require medium pressure and medium shear for cell rupture. And the yeast cells are among the toughest cells to be broke. Usually, to rupture yeast cells, you would need the highest amount of pressure and the highest amount of the shear, and also sometimes you have to use multiple passes to achieve a high rupture efficiency. So the first example is the cell disruption of an E. coli cell. In this test, after we recovered the protein, there's a organophosphate hydrolysis reaction to test for total protein recovery. In this test, the cells were grown in a 2-liter bioreactor. The eruption process were actually performed under different pressures. We used the 3 different pressures. We did 1 and 2 passes, and we analyzed the results by getting the total protein recovery, and also we took some TEM images for visualizing the results. Here on the right are showing the TEM images. You can see the unprocessed cells and versus the image after just one pass. Like I said, the microfluidizer is really efficient. Usually after one pass you can achieve well above 90% of the disruption efficiency. You can see from the images here. As to optimize the process conditions, we can look at the reaction rate, which indicate the total protein recovery. So you can find that the maximum protein recovery is achieved at the medium or the middle pressure we used in this test after 2 passes. After that, if you increase pressure or you increase the number of passes, you will see a decrease in the protein recovery. Possibly means the protein denature have occurred in the process. So this is the optimum process for this type of cells and application. The next example is a disruption of the yeast cell. Like I mentioned before, yeast cells usually require higher pressure. So this test was done at 30,000 psi. You can see after this is the material is 5% yeast in the aqueous phase, and after just one pass, you can see visually majority of the cells are being ruptured, which achieved a pretty high efficiency. This is a different type of yeast cell that were ruptured. This is actually done at a 30,000 psi with our highest shear rate chamber, G10Z, which gives the highest amount of the shear rate. And you can see in this case, the microscope picture indicates we have to use up to 5 passes to achieve roughly 90%, 95% of the lysis rate, and at 10 passes, almost all the cells were broke. But when you analyze remember, you want to rupture as many cells as you can, but at the same time, you want to recover the good product. So when we measure the soluble protein concentration, you can see it reaches to a maximum after 5 passes. So additional passes actually, the concentration will start to decrease, which implies that protein denature may have occurred. So in this case, for this application, under this process conditions, 5 passes is the optimum process condition. Now move on to size reduction of solid suspensions. As I mentioned before, most newly developed APIs are water insoluble, which has very low solubility in water. Because those drugs will stay in the body for a short period of time, if you want to use this APIs to achieve a desired effective therapeutic results, usually you have to administer a large amount of the drugs, which is really bad because a lot of the APIs are actually toxic. So anything that will not dissolve in the body will have to be removed by the liver. I want to point out here is that reducing the particle size of these particles will actually not change the solubility, because the solubility will not change. What will be changed is by reducing to a smaller particles, you're actually increasing the surface area, which increase the dissolution rate. Imagine it's a smaller, a very, very small sphere or particle can be dissolved much quicker than a big particle or sphere. So this all often translates to the increase in the bioavailability of those particles. Example I'm showing here is actually the size reduction of a tolocone suspension. this is what the total cone suspension looks like. It's yellow in color. It is used for treating several different disease, including malaria, toxoplasmosis, babesia, and when treating some of the disease, it's often used in combination with other drugs. Let's now focus on the particle size distribution here. The blue curve shows the unprocessed samples, which shows some of the large particles. Here we present results from two different combinations of processes. One is from a process operated at the lower pressure, which means the lower shear, but with more number of passes. The other one is actually, you can see the two curves are overlaid on top of each other, which means we can adjust the process, for example, higher pressure, which gives a higher shear rate, but using only half of the number of passes. So we can essentially deliver the same results in terms of particle size and particle size distribution with a different route. So it depends on your requirement or need. You can either choose operating the microfluidizer at a lower pressure but doing a little bit more number of passes, or you can achieve the same results at a higher pressure with less number of passes. So depends on your requirement and need, and we'll have a solution for you. Here's an example of reducing particle size of a high blood pressure drug. You can see from the unprocessed sample here, there's some big crystals here. To the particle size distribution are shown here to the right. They're in several hundred micron in the size. And after just one passes up through microfluidizer, you can see the D50 is already reduced to just below 1 micron. So the particle size are reduced to submicron range after just one passes. If you need to reduce this further, you can increase the number of passes to achieve a smaller size. Another example of API suspension, you can see from here, this is a needle-shaped crystals with initial size close to several tens or 100 microns. Again, showing here are results from a lab scale microfluidizer and industrial microfluidizer. You can see they both achieved very similar results. In the lab scale case, the particle size of D50 was reduced to just below 800 nanometers. The production scale machine achieved a slightly better results with a little over 400 nanometers for D50. This is the result of, I mentioned earlier that sometimes you do get additional benefits for production microfluidizer using two intensifier pumps. But overall, you can see the results are very similar, achieved from both lab and industrial scale microfluidizer, which proved the scalability of our technology. Now I want to move on to liposomes. So as you probably all know, that liposomes are spherical lipid vesicles constructed with a bilayer membrane structure. The liposome is versatile to because the unique structure allows it to encapsulate either hydrophilic components inside the core or hydrophobic components inside the bilayer lipid region, or it can encapsulate both compounds. And also, liposome is by far one of the most successful delivery systems currently in clinical use. This is an example showing the creation of liposomes. This was done for a large US-based pharmaceutical company. And it's a liposome formulation for treatment of cancer. The goal of this test was actually to create liposomes in the target range of 50 nanometers to 150 nanometers. The results are shown here. You can see this is the unprocessed samples with some large droplets. And after processing, you can't visually see the particles here because they're all much smaller. The distribution showed that they are within the requirements of this test, and we're able to achieve the desired results, which means create the liposome particles that are with satisfying particle size. My next application is polymer nanosuspensions. So polymer nanoparticles is another system that can be used as delivery system. They're interesting particles because the ability to encapsulate a combination of different drug products. They can imagine you can capture more than one type of drugs inside the matrix. Also, their surface can be easily modified to include targeting moieties, so you can achieve desired target delivery properties. The other thing interesting for polymer nanoparticles is you can encapsulate both the therapeutic payload as well as the imaging payload inside at the same time inside the matrix, which offer you the capability of performing diagnostic as well and the therapeutics at the same time. So the way we use microfluidizer to create these nanoparticles, basically using the emulsion evaporation method. So basically, we're forming a nanoemulsion with the polymer and drug dissolved in the solvent as an oil phase, and then disperse this oil phase into the aqueous phase using the microfluidizer to create a nanoemulsion. After the creation of the nanoemulsion, the solvent is removed either by evaporation or by solvent exchange. By doing this, the nanoparticles will be hardened and form a suspension, and you can put this through suspension in for further post-processing steps as a purification or concentration, concentrate them. So a couple of things I want to point out here is in this method, so you have to be careful that the solvent should not be miscible with water. And also, the steps after creation, the nanoemulsion is also important, such as this solvent removal steps. It can impact the way those particles formation as to affect the particle size and the morphology. So here's the example of creating PLGA nanoparticles. The solvent used in this case is DCM. It's processed for just one pass at 10,000 psi, which is not a very high pressure. You can see the TM images here showed here. The dark big circles represent polymer nanoparticles. The small black dots here are actually contrasting agents added to the solution for enhancing the image. We looked at the effect of the concentration of PLGA in DCM to the particle size. You can see as you increasing your PLGA concentration, you are getting smaller particles, and at a point, if you continue increasing the PLGA concentrations, the particle size will actually increase. So if you want to get, in this case for this formulation, you want to get a smallest particle size, this is the optimum concentration you want to use. So my last example is actually reduce molecular weight of polysaccharide. Polysaccharides are utilized in many pharmaceutical applications because they're natural to our body. They're biocompatible and biodegradable inside the body, and they can be broken down to the body building blocks naturally. This is allow for drug release without inflammatory immune response. Polysaccharides come with diverse molecular weight and structures. They have the hydrophilic groups enable bond formation with tissues and mucosal membranes and extended circulation in blood. So this can increase the probability of targeting of tumors. Their hydrophobic groups can prevent, their hydrophobic groups also prevalent on polymer backbones. So this is a good carrier for water-insoluble drugs. The driving force to reduce the molecular weight of polysaccharide is because the low molecular weight polysaccharides show positive zeta potential and higher solubility at a neutral pH. This is an example that we're trying to reduce a polysaccharide with the molecular weight down to 250 kilodalton. So in this case, in this example, in this test, we tried 8 different types of polysaccharide. And you can show with the molecular weight reduction versus number of passes here. In this test, we're able to achieve the target, which is 250 kilodaltons for 7 of the 8 polysaccharide samples. Some of them actually only require a few number of passes. Some will require a little bit more passes to achieve the desired molecular weight. There's only one shown in the green curve here that did not meet the requirement because the reduction rate is not fast enough. But overall, Microfluidizer is very efficient to reduce the molecular weight of polysaccharide. So with that, I want to conclude my presentation today. So the take-home message really is that Microfluidizer high shear fluid processors can produce nanomaterials with a wide variety of multi-phase applications. I hope the examples I showed obtained with Microfluidizer prove that our technology is superior to conventional technologies. We're very efficient and reliable. The Microfluidizer is able to offer precise, repeatable, and scalable results. Lastly, I want to emphasize that for biopharma industry, the production Microfluidizers are designed for compliance with cGMP and are capable of CIP and SIP operations. With that, I want to thank you for your attention and be happy to answer any questions you might have. I also have my contact information listed here with my phone number and email address. You're welcome to give me a call or send me an email for any additional questions.
Well, I think this brings us to the end of the webinar, and so I sure appreciate your presentation. It was very interesting, and thank you to everybody who came out and listened. And if you have any further questions, go ahead and you can write to us later on, ysu@idexcorp.com, or you can contact us at labinfo@horiba.com. Until next time, bye for now. Okay, thank you very much.
Thank you.